global player with a monopole on the orbit trap. I have it in my lab, and I'm so proud to have that machine that gives you 4 digits after the dot and the sum or molecular formula. Today you have no orbit trap, but you brought with the latest HPLC system. And you, Chris and Monica. Chris, you start. And what is the innovation with this newest system?
Hi, thanks. I'm joined here with my colleague Monica, and we're here to show off the Vanquish LC system and our Charged Aerosol Detection, our latest one, which is the Vanquish CAD P Series. And when it comes to liquid chromatography, generally speaking, most people associate that with UV detection, which we have here. And that's because many compounds have chromophores, so the UV detection is the perfect option for that. That's why probably around 80–90% of the market for LC is with UV detection. However, many new therapeutics coming through, and also many of the impurities associated with them, they don't have chromophores. So in that case, UV detection isn't suitable. And that's where the charged aerosol detector comes into play. It's perfect for compounds that don't have chromophores because it has the ability to detect all non-volatile compounds and many semi-volatile compounds. It gives a uniform response as well, so it makes standard-free quantitation quite easy as well. Now, the CAD has been out in the market for over 20 years now, so it's very well established. And earlier this year, we launched the CAD P Series back in July as well. Generally speaking, you'll find charged aerosol detection across all forms of industry. So from pharmaceutical analysis, like looking at impurities and drug profiling, to biopharma analysis, and then moving to, say, natural products and food and beverage. So with drink analysis, we can look at all the additives that have been are in there, as well as the sugar content and the caffeine content as well. And then you'll also find it in various labs, such as environmental labs as well. But today we're going to be focusing on pharmaceutical analysis, so looking at Polysorbate 80. This is a compound that you'll find in many therapeutics, such as vaccines, such as the flu vaccine, COVID vaccine, along with cancer treatments and other therapies as well. You'll also find it in cosmetics as well. The purpose of Polysorbate 80 is that it's a surfactant and an emulsifier that helps stabilize the compounds. So it'll help increase the shelf life so they don't break down, but they also have an added role that it helps the body absorb the treatment as well. And because of that, because it's been absorbed by the body, it's very important to monitor the impurities and make sure that it's manufactured to within tight standards. That's what we'll be focusing on today, because with Polysorbate 80, it doesn't have any chromophores, so you'll need some kind of universal detector to really analyze it. So Monica will set up our sample, and we'll be analyzing it on our Vanquish LC system. We'll get that going for you. One thing I should remind you all of is, when it comes to chromatography and especially charged aerosol detection, because it can detect all non-volatile compounds, it will pick up any contaminants. So you do need to use the best quality consumables. So the best way of thinking about it is, if you put quality in, you'll get quality out. So that means, using high quality vials, using high quality columns, and also high quality mobile phases as well. So if I get the sequence started up. Now, one of the nice features of the Vanquish LC system is that it has many smart functions, and one of those is the smart startup and shutdown functionality. And that allows the system to automatically start up, purge the pump, switch on the lamps, and then equilibrate the system before automatically starting the run when it's hit the right conditions. And then once the sequence is complete, it can then automatically shut down, preserving your column and then also preserving your remaining mobile phase. So you can save a bit of money by not having the system run or having to come back to the system. So yeah, it can be a really true walk-away type of system. Now, the way the CAD works, or the way the system works, is that we have the pump at the bottom. So that's going to be pumping mobile phase up to the auto sampler. The auto sampler is then going to inject and deliver the sample into the flow stream. That will then pass on to the separation column, and this will separate the different constituents of the sample out. And then this will flow into the charged aerosol detector here. So we've got this line comes up, and then that feeds into the nebulizer. Now, as I said, it's a charged aerosol detector, so we're generating an aerosol. So what this nebulizer is doing is basically something similar to this. So it's creating many fine droplets, and this gets sprayed into the detector, and these droplets will then pass through an evaporation tube, and that evaporation tube is there to remove all the volatile compounds, so essentially the mobile phase. Once it's passed through that evaporation tube and all the volatiles have been evaporated off, it then goes into a mixing chamber. Then in that mixing chamber, we're firing charged nitrogen gas at the particles that have been formed. And this charge, when it collides with the particles, this charge forms around the particles, and then that passes through an electrometer, and we measure the size of the charge that's been applied to the particles. And then that gives us our detector signal. So with the CAD, as I said, it's a universal detector, and you might have come across some other types of universal detector. So it can be put in the same bracket as, I guess, a refractive index detector. But the problem with refractive index detectors is that it's not gradient compatible, and it's got limited sensitivity. It's got very basic functionality. So the CAD is a lot more sensitive and also is gradient compatible, so you can do fast UHPLC methods on it. Another type of detection, ELSD, and that generates a similar type of aerosol, but that uses light scattering to do the detection. Now, the problem with ELSDs is that you don't get really a uniform response. You get a sigmoidal curve, and that can cause sensitivity issues, and you get lower dynamic range with that kind of detection as well. So the CAD overcomes that. You get a nice response curve that's very predictable, and therefore you get great sensitivity, and you get a higher order of magnitude when doing your analysis as well. Now, this type of analysis normally takes about 40 minutes, so because we don't have that, I can switch across to some data that we've previously run, and I can bring up the chromatogram for that. Now, this looks slightly different from a normal chromatogram where you would normally just have a few peaks and you'd have one peak of interest. But because the way Polysorbate 80 is, it's normally a mixture of several hundred different types of different formations of it, essentially. So what we're looking for here is the different peak groups, and here we've got 4 distinct peak groups. So with this type of analysis, what we're doing is looking at the essentially the fingerprint. So the relative areas between these different 4 groups, and from this, when we're looking at different batches and different lots, we can do a comparison, so you can really do some good lot-to-lot analysis and make sure nothing's changing in the manufacturing process, and make sure, yeah, all the impurities are noted and any changes. So yeah, that's essentially the CAD, and you might be thinking, well, could we not use a mass spec for this type of analysis? And you are right, you can use mass spectrometry for Polysorbate 80 analysis, but what we say is that the CAD's a very complementary technique. So you'll see in this particular setup, we've got UV detection and also the charged aerosol detection, so this is a common setup. So you've got UV for chromophoric compound quantitation, and then the charged aerosol detector for non-chromophoric compound quantitation. And then you can also have an additional mass spec, so whether that's a single quad, triple quad, or high resolution mass spec with an Orbitrap, and that can do the identification. So that's normally, a great setup for method development. Because you'll get the CAD doing the quantitation and also the mass spec doing the identification. And as I mentioned at the start, because it has a uniform response, that means you can use standard free quantitation. So if you're looking at impurities or unknowns where you just don't have a standard, you can use a known standard, completely different compound, and use that to quantitate those unknowns and those impurities. So yeah, that's the new Vanquish CAD P series, and thank you for listening.
So thanks to Monica and Chris. And Chris, you said super sensitive. What does it mean?
Generally speaking, with the CAD, you can go down to a low or sub nanogram levels on column, which you just wouldn't be able to achieve.
Yeah, this is super sensitive. Okay, I agree. So great to hear that latest development. Thank you, Chris.